Hello, I'd like to walk you through our Canvas classroom. You have seen how to log in on your student's computer, and if you wish, you could just ask your student to log in on their Chromebook. They will go to the waffle up in the top right corner of their screen, and they're going to scroll down, or you can scroll down until you can get to Canvas. Canvas will be a blotchy red circle, and that's what we're going to start with. So I'll push this open and show you what I'm going to see. Mine is a teacher view, so it'll look a little bit different, but I'll get it into student mode here as quickly as possible. Your student, in theory, is listed in 4 courses for this fall Q1. And so for our purposes of this video, we're going to look for the biology class. The biology class will be a view of a plant cell with a whole bunch of chloroplasts doing photosynthesis in the image. We'll click this open, and then we should fall onto our home page. I'm going to switch this and get this into our student view, so it should automatically populate this field of view. And what you're seeing here is 4 little buttons under my welcome screen, and if you scroll down just the slightest bit, you'll actually see our daily agenda, which automatically updates with our latest and greatest activities for the day. So today is the 11th of September, day 7 in our Biomolecules of Life unit. Our anticipated exam will be next Tuesday. I always reserve the right to push it back a day. It will never come forward. But I think that we are very likely to be ready for our second unit exam on Tuesday of next week. So things you need to know. The first thing is that this is, when push comes to shove, where you should be seeing. This tells you what the students and I did together during the day. If it's too hard to see, you can come down to the 3 buttons and make it a full screen mode. You can also go back in time with these little greater than and less than. Yesterday, your students learned about Edward Scissorhands while we were doing a demonstration about enzymes. Our enzymes were working like scissors to help cut a substrate, which in our case were straws, and the pieces were our products. We were using this to prepare for a lab we did today, which actually used an enzyme called catalase to break down a poison called hydrogen peroxide found in all of our body cells. That poison is broken down by the enzyme/scissors into harmless water and harmless oxygen gas. So this was our very short abbreviated agenda for today. And again, if your student was absent, say last Friday, then you go back in time and it'll take you right back to what we did last Friday. The other thing that I'd like you to see, besides of course the very important agenda, are these buttons. We have our class syllabus up here, teacher contact. Modules is not a very useful button, but our class notebook is. So let's go ahead and take a look at that. We have a spirally bound class notebook. For this course, students will take it home with them every day, but from time to time I do collect it and grade it. That time is coming pretty soon. Let's make this as large as possible, and what we're looking at is a very, very structured set of reference documents and work pages. I'll give out the format. These were some of the forms we used to explain and identify parts of some experiments we saw about sharks and ants, aquatic plants we moved through. There is a why are cells so small laboratory that we did, and we were talking about a cell's membrane. These pieces will give instructions as to what still needed to get done. The My Thinking is something that will populate in the grade book here quite shortly. But your student's notebook should look very similar to my notebook here. I think we're in page 40 at this point, so there could be quite a bit of material that needs to get updated or colored. Most of these pages need some kind of text, graphics, and color for them to be considered complete. We've been going through conservation of matter and conservation of energy, and then we started working on our enzymes. So you'll start seeing enzyme material populating in the notebook as well. But this is a wonderful way of checking to see where your student is in their understanding of their everyday classwork, because it's usually captured in the notebook. We'll go back over to the home button, and one more thing that I do think that we should look at is going to be the assignments button. Though the assignments are not graded in Canvas, the assignments are graded by me, typically by hand, because we do have much of our work in our in our physical notebooks. There are listed assignments that are posted here with information that you can go backwards and take a peek at. So if a student was noticing in their notebook that their notebook page 29 was not complete, they can click over here in their module 2, our current unit, and they can see what that page was supposed to be looking like. It talks about a feedback bit, what we built in the class, our before and after molecules, and then it will complement what you will see in the actual notebook on page 29. So going back and forth between the daily agenda, let's go back to our homepage. Our daily agenda tells you what's happening each day, our class notebook, and then over to our left side and look at our assignments log. That will keep you on target. If there's ever questions, please do just send an email or ask your student to follow up with me first. And if you don't get a response quick enough, then follow up with me and an email, and we can work together to get all of this new process and procedure nailed down. Thank you so much. Have a great day.